Hello students, welcome to chapter 16. We're going to be talking a lot about gene regulation, gene expression in eukaryotes, prokaryotes, things like that. First of all, when it comes to regulation, we'll be talking about transcriptional regulation. So transcribing is the first step, of course, when it comes to transitioning from DNA to proteins. So fertilized egg divides and produces special cells that differentiate into distinct types of cells. So whenever you get to from the egg to a zygote, you have to differentiate that egg into the different types of cells that make up our body, right? So you have housekeeping genes that are expressed in almost all cell types. Regulated genes that may or may not be expressed at any given time or a given cell type. So these housekeeping genes are expressed in almost all cell types. So they have certain genes that get rid of certain proteins that they don't need, or have certain proteins that they do need. So these genes make up all the proteins, these housekeeping genes make up all the proteins that regulate all those proteins that are needed for this specific function. Transcriptional regulation, process that directly control gene expression. So these specifically express and control those genes. Additional controls affect the processes of mRNA, post-transcriptional regulation. So this is after transcription. Its translation into proteins, translational regulation, and the lifespan and activity of proteins. This is post-translational regulation. So over on the side here, you can see all of the different proteins here, all the different housekeeping proteins. You have activators, enhancers, promoters. Remember that promoter has that tata box. Thymine, adenine. In prokaryotic genes, or in prokaryotic DNA, they use operons. This is a cluster of prokaryotic genes and associated regulatory sequences, all transcribed into a single mRNA. This is a transcriptional unit, all in an operon. Regulatory proteins bind to these regulatory sequences. So you have a regulatory protein, some type of regulatory gene, one regulatory DNA sequence is the promoter, right? Site to which the RNA polymerase binds to begin transcription. So it just binds to that operon and begins transcription from there. You can have a repressor protein which prevents the operon genes from getting expressed. So sometimes you just have a little repressor protein that comes in and just stops that from happening. And this can also happen in eukaryotes. It's just a little bit more complicated. And then there are activator proteins, which turns on gene expression. This isn't in this diagram, but there are. Now we'll talk about specific operons. So the three structured genes involve lactose catabolism, LAC-Z, LAC-Y, and LAC-A are transcribed into a single unit with a single promoter. So the LAC operon is negatively regulated by the regulatory protein LAC repressor. This LAC operon, this whole operon right here, is regulated by this repressor, right? If this repressor comes in and hides that gene, it's not going to work, right? That polymerase isn't going to come through and make mRNA. Okay, the LAC repressor inhibits transcription when lactose is absent from the medium. So when lactose is absent, it doesn't really need the mRNA for lactase, right? It doesn't really need to make that enzyme. The LAC operon is inducible operon though, right? It can be induced. If allolactose, an isomer of lactose, comes in, it's the inducer of the LAC operon. It inactivates the LAC repressor and induces the transcription of operon's structural genes. Right? So if allolactose comes in, it's going to unhinge that repressor and it's going to take that out. But in the absence of lactose, there's no allolactose molecules to inactivate the repressor. The active repressor binds to the operator, blocking the transcription of structural genes. So A down here, you can see the transcription is blocked, right? But in B, you can see that allolactose is the inducer, and that binding site for the inducer inactivates that repressor, and it can make that mRNA. And that mRNA comes out, lactose catabolizes that enzyme, lactase. Lactase can break down lactose into whatever sugars that it needs to break it down into. Positive gene regulation of LAC operon. So a positive gene regulation system ensures that the LAC operon is transcribed if lactose is the only energy source, but not if glucose is present in addition to lactose. The key regulatory molecule is CAP, catabolite activator protein, an activator that stimulates gene expression, basically synthesized in an active form. This basically means that if you have glucose present in addition to lactose, you have this other thing called a catabolite activator protein that can stimulate another gene expression, synthesizing lactase in that inactive form. But if glucose is absent, CAP activated by what's called CAMP can bind to that CAP site in the promoter, and can enable the RNA polymerase to already bind, and transcription can proceed. If you have the lactose present and the glucose is low or absent, structural genes expressed at high levels, right? You have that cap right there and the inactive repressor, right? Allolactase is the inducer. RNA polymerase starts at that cap site. See that? And transcription occurs. But if lactose is present and glucose is present, structural genes expressed at very low levels, then you don't have that active cap, and RNA polymerase cannot bind effectively. It all depends on how those genes are structured. Also depends on that glucose level. So there's another one, regulation of a repressible operon. So this example, E. coli is grown on a medium with tryptophan. The cell is not synthesizing it. Tryptophan is a amino acid. It's very important for any cell. So if E. coli is grown on a medium with tryptophan, the cell is not synthesizing it. If the E. coli is not grown on a medium with tryptophan, E. coli has to synthesize it, right? It's one or the other, right? The tryptophan operon has 5 structural genes, right? Tryptophan E through tryptophan A, or TRPE through TRPA, right? That encode enzymes through tryptophan biosynthesis pathway. The expression of the TRP operon is controlled by TRP repressor protein, encoded by the regulatory gene TRPR, synthesized in inactive form, right? We just talked about repressor proteins and how they work. Tryptophan acts as a co-repressor, though. A regulatory molecule that activates the repressor to turn off the expression of the operon. So, let's see how this works. Tryptophan, when it's absent from the media, it must be made by the cell, right? Structural genes transcribed. So you have your regulatory gene at the end, right? Some type of regulatory gene. TRP repressor. If it's inactive, it's not going through and it's not telling the operator to stop, right? So that RNA polymerase binds to the promoter, operator's not telling it to stop, transcription is occurring. If tryptophan is present in the medium, cell uses tryptophan in medium rather than synthesizing it. Structural genes not transcribed. So you have your regulatory gene over here telling the TRP repressor. But the tryptophan is there. It's a co-repressor, right? Binding to that tryptophan binding site. So that's active, telling the operator to stop. So transcription is stopped. Okay, comparing the LAC operon and the TRP operon. So the LAC operon is a repressor is synthesized in active form. So it's already active whenever it's synthesized. And the inducer allolactase inactivates it. The TRP operon, it's synthesized in the inactive form. Tryptophan activates it as a repressor. Also, active repressor blocks transcription of the operon. So whenever it's active, it'll block the transcription. So, regulation of transcription in eukaryotes. So we just talked about transcription in prokaryotes. We could talk about in eukaryotes. Eukaryotes are complex, more complex than prokaryotes because we have a lot more membranes and other things going on, like proteins and such inside of our cells. So nuclear DNA is organized within histones and chromatin. Multicellular eukaryotes produce many types of cells. The nuclear envelope separates the transcription and translation. So our expression is regulated at different levels, right? Transcriptional regulation, post-transcription, translation, post-translation, right? So we have many different levels that we have to regulate. A eukaryotic promoter can exist in two different states. Of course, we've seen this before. We have the inactive state, where the nucleosomes prevent general prescription factors, transcription factors, and RNA polymerase II doesn't bind and doesn't occur. In inactive chromatin, histone tails are not as acetylated, right? These are basically wound together. The histones bind the chromatin together, and they aren't transcribed. Active state, general transcription factors bind to the and RNA polymerase II binds to the promoter, and transcription can occur. Acetylation changes the charge of those histone tails and loosens the association of the histone with DNA. So I can show you this in a little picture later. There's also a little picture in your book. Anyway, large multi-protein complexes bind to displace the acetylated nucleosomes in the promoter region of the DNA, or move them away from the promoter. So this is called chromatin remodeling. If you have a large complex of protein and DNA, and the DNA is being moved from one place to another, that's chromatin remodeling. Also, there's a histone code. This is a regulatory mechanism for altering chromatin structure and gene activity based on the signals on the histone tails represented by chemical modification patterns. Basically, you have the histones are binding and winding all the DNA based on their chemical structure, their chromatin structure, and it's all in line with how the patterns are formed. Okay, this is what I was going to show you. So if the nucleosome is an inactive chromatin, is basically hugging that DNA, that histone is hugging that DNA, and you can see the histone tail is just kind of binding it. But if it's acetylated, you can see the tail is kind of winding out. So that's an active chromatin. And that gray is the DNA. So you can see it can kind of get loose there. DNA methylation. This is where enzymes can add a methylated group, CH3, to cytosine bases in the DNA. This is important because they can silence the promoter regions. You basically turn off those promoter regions in DNA transcription. Another area to control transcription. This is an example of what's called epigenetics, in which a change in expression does not involve a change in the DNA sequence. It's just changing it chemically. DNA methylation underlies events such as X chromosome inactivation and some genomic imprinting. This can happen randomly, it can happen non-randomly, just depends. Okay, the promoter immediately upstream of the transcription unit contains sequence elements such as the Tata box. Remember that Tata box? It's in the promoter. Transcription factors, right? We need these transcription factors to recognize and bind to that Tata box to recruit that RNA polymerase. Upstream from the promoter, we have the proximal region and some proximal elements. Even further up is the enhancer region. In that region, we have regulatory sequences where regulatory proteins can bind to it. This is kind of like what we saw in prokaryotes, but it's a little bit more complicated than that. So these regulatory proteins can stimulate or inhibit transcription initiation rate. So activation of transcription, general transcription factors, these are going to bind to the promoter region, right? Transcription initiation complex, you're going to have a bunch of these transcription factors, these transcription proteins. Basically there to make sure that the RNA polymerase binds to the DNA. Activators specific to each cell type bind to the promoter proximal elements. I want to say specific to each cell type because sometimes those cells are completely different from each other. They interact directly with the general transcription factors and stimulate transcription initiation. Remember what I was saying earlier, that prokaryotes don't need these transcription factors because they don't have different cell types that need this. Multicellular organisms need this because they have different cell types that require these transcription factors in order to activate RNA polymerase to go through and create RNA. A large coactivator forms a bridge between the activators and the enhancer. Remember that from the previous slide. And proteins at the promoter and the promoter proximal region causing the DNA to form a loop. So that's this next slide. So the enhancer comes through. It's a little bit far off from it, but the coactivator and the two activators on each side forms a loop, right, to bring them together. And all those proteins together help that RNA polymerase to get going to make RNA. See how this is way more complex than prokaryotes. In some genes, repressors oppose the effect of activators, blocking or reducing the rate of transcription. So some repressors will bind to the same regulatory sequence to which activators bind. So those activators, sometimes repressors will stop that binding from happening, that loop from happening. Sometimes repressors bind to their own specific site and interact with the activators so it cannot interact with the coactivator. So there's a lot of things that can happen to stop that loop from happening. Some repressors bind and recruit co-repressors that inhibit transcription initiation. Combinatorial gene regulation. So the combination of a few transcription factors in particular ways controls the transcription of a wide array of protein coding genes. So if you have like different genes in the enhancer, you have multiple activators required to activate that transcription of a particular gene, right? You have 3 activators in this enhancer in order for this one to work, this gene to be transcribed. So there's a lot of stuff that can happen if this one gene is to work. Okay. Coordination of genes with related functions. In eukaryotes, genes that must be coordinated at all times have the same regulatory sequences. One signal controls the transcription of all these genes simultaneously. For example, steroid hormones. A steroid hormone acts only on a specific target tissue that have a steroid hormone receptors in their cytoplasm. So one single steroid hormone regulates all the genes that have the same DNA sequence. Basically, if you don't have that steroid hormone, all of those genes don't work. And that's an issue. Which is why a lot of people, if they don't have certain steroids, if you don't have certain hormones, it's not good. So we have the steroid hormone moving through the cytoplasm in the membrane. It's binding to a certain receptor, and it has to get through that membrane. Once it gets through that membrane, it binds to the promoter region or an activator region. It has to activate that region in order for it to get to the RNA polymerase. If it doesn't activate that region, or if that protein is somehow mismanaged, that RNA polymerase doesn't work, the gene isn't controlled, somehow the gene doesn't work, that pre-RNA isn't working, that protein isn't working. There's a lot of proteins that use that steroid hormone. Okay. Now we're going to talk about post-transcriptional regulation. Pre-mRNAs, after it comes out of the transcription, can be processed by alternative splicing, right? Not through SNRPs. So producing different RNAs by removing different combinations of exons along with the introns, right? Regulatory molecules such as the steroid hormone may be indirectly or directly slowed to increase the effects of mRNA breakdown, right? And then there's RNA interference. This is non-coding single-stranded RNAs binding to mRNA and affecting their translation. Two major groups in this: microRNAs and short interfering RNAs. MicroRNAs are transcribed from mRNA genes, miRNA genes, and then short interfering RNAs are produced from double-stranded RNAs not encoded by nuclear genes. This is kind of weird because they kind of fold in on themselves. It's just, this is a weird process. So microRNAs, this is a protein complex that binds the sequences in the 3' UTRs of a target. So if miRNA and mRNA pair imperfectly, the target mRNA is not destroyed. The double-stranded segment formed between the two blocks of ribosomes, they stop from translating, right? They basically form together. They pair perfectly. An enzyme of the protein complex cleaves the target where the miRNA is bound to it and destroys it. Keeps it from expressing. It sees it as foreign and keeps it from expressing it. And then there's siRNAs. So sometimes these cells are attacked by an RNA virus that has double-stranded stage. So sometimes these siRNAs come from another body, like a virus, which they produce from, their cycles. Double-stranded RNA is cut into short double-stranded RNA molecules and degraded by a protein complex to create single-stranded RNA RNA-induced silencing complexes. So these will attach themselves to mRNAs and just basically silence the mRNAs. Okay, translational and post-translational regulation. Translational regulation is involved by the cell cycle, controlled in all eukaryotes, and in many processes during development in multicellular eukaryotes. So the number one thing I'm going to say here is it involves adjusting the length of the poly-A tail of the mRNA. So just increasing the length of that poly-A tail basically decreases translation. Okay, post-translational regulation controls the availability of functional proteins in 3 ways. So you have chemical modification, additional removal of chemical groups can alter protein activity. Some processing proteins are synthesized a different way. Inactive precursors, activated control, removal of a segment or whatever. and then degradation. These are by proteosomes. So if you have some type of proteasome or some type of protein and it goes through a proteasome, it's just going to degrade it completely. Please give me the type of histones involved in DNA binding. Okay? I know that there is a complex of histones in DNA binding. Please give me the types of histones involved. Okay? I know that's probably a question that's going to come up. So, all right, see you later.